Do I see it's recording? It's here, right?
Yeah.
Okay, good.
Thanks, Trevor.
Thank you.
Thanks, Dr.
Good
Rojas.
afternoon, everybody. My name is Tibor Rohács. I'm giving the renal lectures almost exactly 2 years ago. This was the last lecture I gave in an auditorium. The pandemic hit right in between the first 3 lectures and the second 3 lectures, if I recall correctly. This is my first lecture since the pandemic that is live, even though it's kind of mixed with the Zoom system. I hope you don't mind if I don't wear my mask. I'm not sure how the audio will come out with the mask. You're far away enough. I think it should be safe. I'm triple vaccinated or whatever. So I'm going to talk about the kidney. There's something on my screen. It's on yours, right? Too. Okay, so let's start with the simple question of what do the kidneys do? So the kidneys are the organs that generate urine. But the point really not is to generate urine, but through generation of the urine to regulate body fluids and electrolytes and minerals. So here's a short list of things that the kidney regulates. The idea is if you have too much water, then in your body, the kidney can excrete water to return to normal water levels. And the same thing is true for sodium, potassium, calcium, phosphate. Whenever an electrolyte concentration or amount in your body goes up or down, the kidney will change its excretion to the urine to compensate for those changes. That is number one function. The second function is removal of waste and toxic substances. Again, this is still within the function of generating the urine. The difference between the first point and the second point really is that in the first point, you have a clear set point. You want to regulate sodium around 140 millimolars per liter, whereas with waste and toxic substances, your set point is 0. You just want to get rid of them. Things that you don't need, the kidney will very efficiently dispose them into the urine. Point number 3, which is often discussed separately, is acid-base balance. We're going to have roughly half a lecture about the kidney's contribution to acid-base balance. Again, this is still within, you can also place this function within the context of the first point, because what you do here is you regulate the free proton concentration when you regulate acid-base balance. The kidney is not the only organ that regulates acid-base balance. It's basically a nice collaboration between the kidney and the lungs. And finally, function number 4 is independent of generation of the urine. It just happens so that there are 3 hormones that the kidney makes. Those are erythropoietin, active vitamin D3 or calcitriol, and renin. We will briefly talk about this, but in more detail, you will have at least some of these also in the endocrine lectures. So how do the kidneys work? So on the macroscopic level, there's one tube that goes in and there are 2 tubes that come out. The one tube that goes in is the renal artery, where the blood supply goes into the kidneys. The kidneys has very high level of blood supply. That is important for the kidneys to function properly. And there are 2 ways for the fluid that goes in to leave the kidney. As for every other organ, you have the vein, where much of the blood that went in will come out. But with the kidneys, you also have a second outlet, which is the ureters, and that's where the urine leaves the kidney. And the idea is that if you have too much sodium, then instead of all the sodium going back to the renal vein, some of it will be deposited into the urine. If you don't have enough sodium, the kidney will try to retain as much sodium as possible, and your urine will have very little sodium. So that's how it looks in the macroscopic level. And I try to find a good motto for the kidney, and this is I think this kind of tells you what the kidneys do. Saying the product of the kidneys is urine is like saying the product of a factory is pollution. Urine is a byproduct, and the product is homeostasis. And what is homeostasis? There's a nice definition here. Any self-regulating process by which biological systems tend to maintain stability while adjusting to conditions that are optimal for survival. If homeostasis is successful, life continues. If unsuccessful, disaster or death ensues. So if you don't have kidneys, you die. But of course, today we have nice therapies and kidney replacement, dialysis. So it's not necessarily true. Where does this motto come from? It comes from Twitter. You can't only you can follow celebrities and politicians, but you can also follow scientists and physicians. And this is from Joel Toff, whose Twitter handle is Kidney Boy, who tweets quite a bit about renal physiology and electrolyte balance. So the lectures in which we're going to discuss the kidneys are going to be the first 3 lectures today: introduction, body fluids and clearance, that is this lecture. In lecture number 2, we're going to discuss the first step of kidney function, which is glomerular filtration. And in the 3rd lecture, we're going to discuss sodium balance. Then next week, we will come back to water balance. And the last 2 lectures, 5 and 6, where we discuss smaller topics like reabsorption of nutrients, potassium, calcium, magnesium balance, secretion, and bicarbonate reabsorption. So you saw this picture before about how the kidneys work on the macroscopic level. Blood goes into the artery and comes out either the vein or the ureters. At the microscopic level, the functional unit of the kidneys are the nephrons. And what happens in the nephrons is step number 1 is filtration, where the kidneys filter an enormous amount of fluid, 180 liters per day, which is 20% of how much blood or plasma goes through the kidneys. That's going to get filtered. And when we mean filtered, protein-free plasma is going to enter this tubular system with basically the inside of the nephron. And of course, nobody ever loses 180 liters of fluid per day. So the vast majority of that fluid that was filtered by the kidneys is reabsorbed. So reabsorption is the process where the fluid that enters the tubular system will get moved back first to the interstitium and into the general circulation. So this is reabsorption, when fluid moves from the tubuli to the interstitium and back to the blood. There's an opposite movement, which is secretion. Quantitatively is much less important. You filter 180 liters and you reabsorb 179 liters. So this is quantitatively important. But certain things like potassium and protons are moved by the kidneys the opposite direction. It's moved from the interstitium and from the blood back into the lumen of the tubuli. For most things, the regulated step is reabsorption, for example, for sodium. But certain things, the regulated process can also be secretion. And the examples for this are potassium and protons, as you will see in later lectures. And finally, the last step, where the filtered fluid, after modified by reabsorption and secretion, leaves the kidneys as urine, is the excretion. So excretion and secretion sounds quite similar. Don't confuse them. So remember, excretion is whatever goes out to the external world as urine. So this is the simplest picture I could ever get of a nephron, but a nephron doesn't look like this. The reason why picture number 1 of the nephron is this, because it's very simple to conceptualize these 3 different steps plus excretion. In reality, in mammals, the nephron looks like this. It has this strange loop structure. And let's see if I can use this. It doesn't allow me to annotate. Anyway, so it has this interesting loop structure, which is called the loop of Henle, where the first step in the tubular system from the Bowman capsule, where the filtration happens in the glomeruli, the tubular fluid enters the proximal tubules. This is in the renal cortex. These proximal tubules reabsorb a large amount of fluid. They have lots of mitochondria, and they do a lot of active fluid movement and electrolyte movement. And from the cortex, the tubular system goes down into the medulla of the kidney. And this is the descending thin limb the ascending thin limb, and the thick ascending limb. These three structures together are called the loop of Henle. And so this was just a very brief introduction. We're going to go through all these nephron segments, mainly in the sodium lecture, and later on we're going to revisit these different parts of the nephron in each and every electrolyte and the water balance. So the first lecture will be an introductory lecture where we discuss body fluids and electrolyte. In other words, what is it that the kidney actually regulates? Water and electrolyte balance. Water movement is very important, and water in the human body and in any living organism, for that matter, is moved by osmosis. So osmosis happens when you have two different fluid compartments separated by a semi-permeable membrane. So water movement is free in between these two compartments, and you have impermeable substances in one side and the other. These are the solutes. And if the concentration of these impermeable solutes are different on one side and the other, then what happens, water will move from one side to the other. This is osmosis. And basically equilibrating the concentration of two sides. So the water movement is dictated by the concentration of the impermeable substances. And this is what happens when osmosis takes place. Water will move from one side to the other, and this water movement will happen until the concentration of these substances that the membrane is impermeable to will be equal on the two sides. And there's a reason why water moves this way, because the concentration of water is very high. That's the solvent. So the vast majority of molecules in an aqueous solution is water molecules. So it's really hard to design by evolution some kind of a pump mechanism which will take water molecules one by one. It's much easier to move the impermeable substances from one side to another because you have just simply less of them, and water will follow it. So there's an internal logic why water always moves with osmosis. So osmotic concentrations can be defined in two different ways. They're very similar to each other. So osmolarity is the number of particles per liter solution, and it is expressed as milliosmoles per liter. Osmolality is the number of particles per kilogram solvent. And in case of relatively dilute solutions, these two are very close to each other. So most people use this interchangeably. And there's a third way to express osmolarity is tonicity, which refers to the hydrostatic pressure that the osmotically active particles exert. The reference point in renal physiology will always be the plasma osmolarity. So when I say isosmotic or isotonic, I always refer to the osmotic concentration that equals that of the plasma. And most physiology textbooks and my lectures will use these concepts interchangeably. So when I say isosmotic or I say isotonic, it's basically the same thing. So normally when we talk about osmosis, the most important osmotically active particle in physiology will be sodium, but most other small electrolytes that are impermeable also contribute to osmolarity. But there's a different kind of osmolarity which happens, which is exerted by proteins, because the cell membranes are not permeable to sodium and not permeable to small electrolytes, but the capillary membranes are permeable to sodium, glucose, you name it, everything that's small, but they're impermeable to proteins. So colloid osmosis or oncotic pressure is exerted by proteins. You must have had this as Starling forces from the cardiovascular lectures, but I kind of reiterate because we will operate with this concept also at kidney physiology. So the major body fluid compartment where water is located is the intracellular water and the extracellular water. So roughly 60% of our body weight is water. So the total body water is 60%. It's 42 liters for a average size 70 kilogram male. The intracellular fluid, 2/3 of this is intracellularly located. 40% of the body weight, or 28 liters, is intracellular fluid. And the extracellular fluid is divided between the interstitial fluid, which is basically the fluid that's in between your cells, in your muscles and in other tissues, and between the blood plasma, which is the cell-free part of your blood. 75% of the extracellular fluid is interstitial fluid, and 25% is in your blood plasma. And the blood, of course, is constituted from the plasma and the red blood cells. And the relationship between these can be described by the hematocrit ratio, which is basically the red blood cell volume and the blood volume. Roughly, on average, is about 0.45 or 45%. And so these are the major compartments where water is located. And so what else is in these compartments? So this slide describes that the extracellular fluid and the intracellular fluid has quite different ionic concentrations. The extracellular fluid has high sodium concentration, somewhere around 140 millimolar, and the intracellular fluid has much lower sodium concentration. Potassium, on the other side, on the other hand, is reversed. So you have relatively low potassium concentration outside and relatively high potassium concentration inside the cells. Why is this? What generates this concentration difference between potassium and sodium inside and outside? It's a sodium potassium ATPase. So basically, the sodium potassium ATPase is a very ancient enzyme. It's basically found in essentially every cell type. It just pumps sodium out, potassium in, and it has a lot of different consequences for electrical excitability, osmoregulation, potassium regulation, etc. So chloride is usually the counter charge for sodium outside. Inside, you have a lot less chloride because you have more proteins and you have more phosphate. And I also mentioned calcium here, which is around 1.1, 1.2 millimolar free calcium, and you have a lot lower calcium inside, and that's how calcium can act as a second messenger. And the osmotic concentration between the two sides, it's always equilibrated because most cell membranes, with some notable exceptions, are permeable to water. So water always equilibrates between the intra and the extracellular compartment. So this is the intra and extracellular fluid. And among the extracellular fluid, the plasma and the interstitial fluid is mainly different in the concentration of proteins. You have more proteins in the plasma than you have in the interstitial fluid. And the boundaries, again, the cell membrane is the boundary between the intracellular fluid and the extracellular fluid. It is impermeable to electrolytes, but permeable to water. And the capillaries are the boundaries between the extracellular fluid, between the interstitial fluid and the plasma. And this is impermeable to proteins and permeable to everything else. And this is where colloid osmosis is dictating water movements. So intake and output of water. We're going to talk mainly about regulation of the volume of urine, which is usually roughly equal to how much we drink. But drink is also regulated by thirst. These are the two main things that regulate water balance. And of course, you have other sources of water. Your lungs, your skin, and your feces also excrete some water. And these can also change. If you exercise a lot, your lungs and skin will excrete more water as water vapor. If you have diarrhea, you use more with the feces. And don't forget that some of the end product of metabolism is not only CO2, but also water. And most foods that we consume also have some water. But again, the two key things we're going to talk about is the regulation of the urine, and we will talk just a very little about the regulation of thirst in the context of kidney physiology. So the Darrow-Yannet diagram is a convenient way to plot the osmotic content and the osmotic concentration and the volume of the extra and intercellular fluid. It's a common thing in many physiology textbooks. Basically, the way it looks, you have the ECF, or the extracellular fluid compartment, and the ICF, the intercellular fluid compartment, and the x-axis is the volume, 14 liters for the ECF, 28 for the ICF, and the sort of y-axis is the osmolarity. We simplify for this these slides the osmotic concentration to be 300. In reality, it's a little bit less than 300 milliosmoles per liter. And we're going to go through a couple of changes in this Darwinian diagram when you have different changes in your electrolyte balance. The first example that we use this Darwinian diagram for is an example when you have an isotonic volume expansion. Again, isotonic means the same as isosmotic. It means you take in some fluid that has the same osmotic concentration as the plasma. It's relatively hard to achieve. The simplest way is to infuse IV fluid into somebody. This is an exaggerated version. I think very infrequently get 2 liters of intravenous normal saline, but it's a relatively easy example. So if you have 2 liters of extra isotonic volume infused into you, because the osmotic concentration is the same in the fluid that you receive and your plasma and your intercellular fluid, basically all of this fluid is going to stay in the extracellular fluid. You infuse 2 liters of isotonic saline, osmotic concentration doesn't change, so there's no extra movement. All of it stays extracellularly, and you basically just extend one side of this Darwinian diagram, make it longer this direction, everything stays there, no change in osmotic concentration. Make sense? Yes. No, the idea, thank you for the question. Ask questions because if it confused you that I didn't explain well, so let that be the. So the idea is that the cell membrane in the first approximation is not permeable to sodium or whatever provided the osmotic concentration. So it won't go inside because the solute doesn't go inside the cells. So the solute stays outside and it keeps the fluid outside. Make sense? So the next example is going to be a little more complicated, and I'm going to go to an example calculation from which you can see that if you come up with numbers of how much fluid and how much salt you take in or lose, you can actually calculate these volumes based on relatively simple principles. So what is shown here is an example where you go out to run or bike on a hot summer day, you sweat 2 liters, and let's assume that the osmotic concentration of the sweat is 200 milliosmoles per liter. There's less salt in sweat, but I exaggerated the example so that the changes are larger and easier to appreciate. So you lose 2 liters of water and you lose 200 milliosmoles per liter solution, largely sodium, from the extracellular fluid. So we can go over the calculations of what the final osmotic concentration, the final volumes will be. You can't follow, you can rewatch the lecture, look at the numbers again. So the idea here is that first, we are going to calculate the total osmoles left in the extracellular fluid after you lost 2 liters of sweat. So what you can see here is that originally, the total osmoles were 14 liters times 0.3 osmoles, because we're going to calculate with this simple 300, and instead of calling it 300, we call it 0.3 as osmole. And what you lose is 0.2 osmoles per liter times 2 liters. That's what we lost. And the end result is, if you add up the numbers, you had 4.2 osmoles minus 0.4, you end up with 3.8 osmoles in the extracellular fluid. The intracellular fluid won't change because any change that happens always happens primarily in the extracellular fluid. That's where sweat is generated from. That's where the fluid that you drink goes to. So the intracellular will be 0.3 times 28 liters. It's 8.4. The total volume is the next that we calculate. And so you had 14 and 28, you lost 2 liters, so the end result will be 40 liters. And you can calculate the final osmolarity just adding up the two total osmoles, 3.8 plus 8.4 osmoles divided by 40 liters. So you're going to have 0.305 osmoles per liter, which is slightly hyperosmotic because it started off with 300, just to calculate the round number. You lost something that was hypotonic or hyposmotic, which was a 200 milliosmoles per liter. It's quite logical that the final osmolarity will be somewhat higher because you lost more water proportionally than salt or osmotically active particles, right? Now, if we know what the final osmotic concentration is and the final volume is, and how much we have in one side and the other, we can back calculate the volumes. Basically, what you do is concentration equals amount divided by quantity divided by volume. So if you rearrange the equation, you can get the volume as quantity divided by concentration. And you can see that the new extracellular fluid volume will be 3.8 divided by 0.305, 12.46 liters, and the new ICF will be 27.54 liters. And the conclusion here is that even though all of the 2 liters that you lose first comes from the extracellular fluid, the fact that your final osmotic concentration will be higher, that means that the higher osmotic concentration extracellularly will attract some water from inside your cells, so you lose a little less volume that you would have lost if this whole thing was isosmotic. Does it make sense? You lost a little bit of salt, you also lost water, your extracellular fluid became more concentrated. Some of that water was replenished from basically osmotic forces pulling water out from the intracellular compartment. And if you plug this back into the Darrow-Yannet diagram, this is how it looks. The initial is marked with the dashed line, and the final is the colored one. You can see that the osmotic concentration became higher, both intra and extracellular fluid, because they equilibrated, and the extracellular fluid became lower volume, but some volume is also lost from the intracellular fluid because it moved out to the extracellular fluid. Now, what we do next is, in this imaginary example, you went out for a nice bike ride, it was really hot, you lost 2 liters of sweat, you go home, you get thirsty, you drink 2 liters of water. So you replenish that sweat, which had sodium and electrolytes, with just simple water. And if you redo this whole calculation, I'm not going to go one by one, just do the same as before. The total osmoles don't change. The volume will go back to 42 liters because you drank 2 liters. And if you calculate the final osmolarity, it will go down to 0.2905 liters, which makes sense. You lost sodium and water. You replenish the water. The end result is you just lost sodium. It's just difficult to lose sodium without losing some water first and replenishing it. So your final osmotic concentration will be lower because you lost sodium. The interesting part comes when you look at the final volumes. If you use the same principles as before, you can see that the new extracellular fluid volume, after you lost salt and water, replenished the water, your new extracellular fluid volume will be less than it was originally, even though your total fluid volume is the same. So the take-home message from this little double exercise is you lose salt from the extracellular fluid. And if you lose that salt from the extracellular fluid, what happens, what happens is some of that water from the extracellular fluid will go inside the cells because there's more electrolytes there. So if you lose sodium, you lose also volume because osmotic forces will move the water to inside your cells. Does this make sense? Many of you nodded, so I hope it does. Theoretically, yes, you have to exercise quite a bit to have a significant swelling. But basically, if you exercise and all you do is drink water, you get a little bit of swelling. This was a badly exaggerated example. Sweat is a lot less. Sweat has a lot less salt. But this is why if you go to the New York City Marathon, then every second stand has the Gatorade. So make sure you drink salt, not only water, because basically, yes, you can get hyponatremic. And if we plug this into the Darwinian diagram, this is what happens. Total volume is the same, and basically water moved from the ECF to the ICF, making the extracellular fluid less in volume. I use another example, a slightly different one, with the same end purpose to demonstrate the idea is that the osmotic forces dictate water movement, and one of the key messages from this lecture will be that your sodium content is a very, very important determinant of your fluid volumes. And what this one shows is this intravenous fluids, where the idea is that we infuse somebody with 1 liter of different fluids with different compositions, and we will see how much of that fluid goes intra and extracellularly. So the first example is 5% dextrose, which is basically glucose, and the idea is that you should not infuse anybody with distilled water because that's going to lyse the red blood cells. But if you infuse somebody with something that's isosmotic but gets metabolized very quickly, it's essentially the same as infusing water. Or I could have put drinking 1 liter of water here, which is basically the same. So what happens is if somebody is drinking purely water or gets something that gets quickly metabolized, and it's the equivalent of drinking water, then 2/3 of the water will go to the intracellular fluid and only 1/3 stays outside. Because if you simplify this with just water, if water goes to your extracellular fluid, it's going to dilute it, and much of the water will go inside the cells until it equilibrates. And since the intracellular volume is twice the extracellular, usually twice as much goes inside than stayed outside, right?
For the last part, the osmolarity goes down because we weren't able to replenish the pulse from the part B, right?
Yes.
Okay. So the water is being pulled into those cells because there's low salt concentration inside?
The water will get inside the cells because you lose sodium from outside.
Oh.
So the inside the cells, you have the original osmotically active stuff. It's not sodium, it's mainly potassium. But basically the idea is that you drop the extracellular osmolarity, then water will move inside until it equilibrates. The next example here is when you take some of the dextrose out and put a little bit of salt, you can see that somewhat more goes inside than outside. And if you go half normal, then again, somewhat more stays outside than inside. And basically, if we get to the last example, which is normal saline, which was the very first example, then all of it stays outside because it's isosmotic. There's no reason to go inside the cells. All these exercises serve the purpose of what is shown on this figure is to try to bring home this message that the primary determinant of your extracellular fluid volume is your sodium content. Sounds somewhat paradoxical because volume is provided by water, but you will see throughout several lectures, you're going to deduce all this, that really on the long term, your extracellular fluid volume is largely determined by how much sodium you eat, not so much how much water you drink. And the reason is because it's the osmotic activity of sodium that keeps water outside. And one example we discussed, if you just drink, much of it goes inside the cell. But of course, if you drink too much, eventually you're going to also excrete it with your urine. So that's another reason that you just can't drink yourself to hypervolemic. And on the other side, if your sodium content decreases, again, the end result usually will be hypovolemia. And so the sensors that regulate sodium regulation are primarily volume and pressure sensors. And as you will see in the last lecture today, the major regulator will be the renin-angiotensin 2 aldosterone system. On the other hand, water regulation is mainly regulating your osmotic concentration, not so much your volume. So if the water content of the extracellular fluid becomes high, then the end result is hyposmosis or hyponatremia. And if the water content decreases, the end result is hyperosmosis. There will be some modification of this basic principle, but let's start with the simple message first. And so the sensors of water reabsorption are primarily osmosensors, and the regulator will be ADH, as we will discuss next week. Yes?
When you say hypervolemia here, that means the volume of the ECF is hypervolemia compared to the
Thank you for asking this, because whenever I say hypervolemia or hypovolemia, it always refers to the volume of the extracellular fluid. And the reason why that is very important to regulate, because the volume of your extracellular fluid will be the determinant of the volume of fluid inside your blood vessels. And that will be one of the determinants of your blood pressure, which you already had from cardiovascular. So one of the major functions of the kidney really is the regulation of your blood pressure. If you have high extracellular fluid volume, the end result will be high blood pressure. And if you have very low extracellular fluid volume, the end result could be low blood pressure or even shock. So the major reason we regulate sodium is to regulate your blood pressure, basically. So the last part of this lecture will be the clearance principle. It's kind of a jump from these fluid volumes, but it fits well, this introductory lecture. And so clearance, let's start with the definition. So the clearance is the volume of plasma cleared from a given substance in a given time. Let's look at the formula. C is clearance. U is concentration of a given substance in the urine. P is the concentration in the plasma, and V dot is the urine flow. So to understand this, let's put in some imaginary numbers into this equation, and then hopefully it will clarify. So how much is clearance if the concentration of substance X in the plasma is same as in the urine? Clearance will equal to V, which is the urine flow. So if this number is the same as this one, we can just get rid of it, and C will equal the urine flow. In other words, if the concentration of something in the plasma is the same as in the urine, you clear from that substance exactly the same volume as your urine is, right? If you have the same amount in the urine than in the plasma, then you cleared, if urine flow, let's say, is 1 milliliter per minute, you're clearing 1 milliliter per minute from that substance, right? What if you have 10 times more in the urine? If you have 10 times more in the urine, you're clearing 10 times the urine flow volume, right? Because that original substance was 10 times in the volume of how much urine you produce in a given time. Does it make sense? Not too many nodes. So so let's go back to the definition. So the definition is a volume of plasma cleared from a given substance in a given time. It's a virtual volume. It's not, you know, a chunk of plasma that's going around. It's a virtual volume of how much plasma that certain stuff was in. And the reason for this definition to be needed because it's independent of the concentration. So if you have something very high concentration in your plasma, it's difficult to compare the clearance of that with something that has a low concentration. So in a way, clearance is something to normalize to the concentration of something. And instead of saying, oh, I'm getting rid of ½ kilogram of sodium today, instead, you're saying that you're clearing an X volume of your plasma from that substance, right? That's why people came up with the idea of clearance, because this way you can compare apples to oranges by basically just referring to the volume of plasma that you cleared from it. Okay. This is one way to deduce clearance, and in past years, some students didn't like it. And so I pulled out another definition, another deduction from a physiology book. Some people like this one better. It doesn't matter. As long as you remember the formula and knew what it is, it's fine. Either the previous slide or this one. So another way to basically derive the clearance is if you take the principle that whatever went into the kidney in the arterial blood has to come out as venous output plus urine output, unless the kidney metabolizes that compound, right? So the amount in the arterial blood should be the same that leaves the kidney in the renal vein and in the ureter. So if you write this as an equation, then the concentration of X in the arterial blood times the renal plasma flow on the arterial side should be equal to the concentration of X in the venous blood times the renal plasma flow on the venous side, which is almost the same, plus the concentration of X in the urine. It's a relatively simple equation. What goes in must come out, or what comes out must go in at one point. And the kind of conceptual leap to get to clearance from this equation is the idea is that in a thought experiment, you replace the renal plasma flow with a virtual renal plasma flow. In other words, a volume that is totally cleared of that substance. So you make this sort of quantum leap, and you say you only perfuse the kidney in this thought experiment with the volume that it completely clears the plasma from, then your venous output will be 0 because you completely cleared it, right? So this becomes 0, and then you end up with P times C equals U times V. And if you solve this for C, that becomes the clearance, because the definition of clearance was the virtual volume that you cleared from that plasma, right? This RPF became a virtual RPF. You only perfuse this imaginary kidney with the volume that you completely cleared, which is exactly the clearance. And then you just have to solve this equation for clearance. So that's another way to derive the clearance equation. Again, what you need to remember is the formula and the definition. It's a virtual plasma volume that is completely cleared by the kidney in a given time. So what is this good for? So the most important practical application of the clearance principle is that you can use it to measure experimentally the glomerular filtration rate. The glomerular filtration rate is the amount of fluid that the kidney filters in a given time. It's a very important kidney parameter, because if your kidneys are failing or not working well, then your glomerular filtration rate will go down. That can be measured or can be estimated. We will dedicate significant amount of time to how to do that. The principal method that was developed many years ago is based on the idea that if you measure the clearance of a compound that is completely filtered and neither reabsorbed nor secreted, then the clearance of that substance will be equal to the glomerular filtration rate. Because if you don't reabsorb and don't secrete, whatever was filtered will be cleared, right? So the clearance of that substance will be equal to the glomerular filtration rate. And such substances do exist. Inulin is a polymer of fructose. It's relatively large, but not large enough. It's not large enough to be withheld by the filtration barrier. So it's freely filtered, but it's large enough not to have any mechanism that will be able to reabsorb it. So if you measure the clearance of inulin, that basically gets you the glomerular filtration rate. We'll come back to the inulin clearance on the next lecture one more time and derive it slightly differently from this. You can basically measure the clearance of anything. So in the case of inulin that's not reabsorbed and not secreted, the clearance is equal to the GFR. For anything that's reabsorbed, the clearance will be less than the GFR. And there are several compounds in your blood plasma that are fully reabsorbed. One classical example is glucose. Glucose is a nutrient. It's small, so it's filtered, but if you need it, you don't want to lose it. So you completely reabsorb, and the clearance of glucose under normal circumstances is 0. If somebody has very severe diabetes, you can have non-zero glucose clearance, but normally it's around 0. And sodium is another example. The clearance of sodium is always between 0 and a lot less than the GFR, because most of it you get reabsorbed. Basically anything that's freely filtered and to some extent reabsorbed, the theoretical range for the clearance is between 0 and the glomerular filtration rate. And the last thing that I mentioned here, where the clearance principle can be used to measure a kidney parameter, is the determination of the renal plasma flow, which can be done with this famous compound called para-aminohippuric acid, or PAH. Para-aminohippuric acid is something that the kidneys secrete very, very efficiently. So basically what happens here, para-aminohippuric acid is filtered just as well as inulin. It's not reabsorbed, just like inulin, but what happens to para-aminohippuric acid, and the details we will come back in a later lecture how this happens, is part of the detoxifying mechanisms of the kidney. Para-aminohippuric acid, if you infuse it at a low enough concentration, it will be secreted from the peritubular capillaries with very, very high efficiency back to the tubular lumen. So basically what happens is the kidney almost completely clears the plasma that goes through the kidneys from para-aminohippuric acid. And therefore, if you measure the clearance of para-aminohippuric acid, it will be very close to the renal plasma flow. Not exactly the renal plasma flow, because there are a small number of the blood vessels in the kidney actually bypass the capillaries. So you need to put in a 10–15% correction. But basically, para-aminohippuric acid can be used, if you measure the clearance carefully, it can be used to measure the renal plasma flow. And that's the end of this lecture. Let's have a 5-minute break.